Hello everyone and welcome back to my channel. In the previous video, we started with the chapter of reproduction in organisms where we studied briefly the meaning of reproduction and its necessity for the continuation of life. We went deeper into the two types of reproduction, asexual and sexual reproduction. We understood the advantages and disadvantages that come with asexual reproduction, so let us once again go through what the characteristics of asexual reproduction are. Firstly, if you remember that it is a form of reproduction involving only a single parent, that means it is uniparental in nature. Okay, then it involves little or no variation. There occurs no gamete formation, and if there is no formation of gamete, there occurs no fertilization. When there is no formation of gamete, no fertilization taking place, then there is no scope of meiosis occurring. And if there is no scope of meiosis occurring, there occurs little or no variation in this form of reproduction, resulting in offsprings which are genetically and morphologically similar, exactly similar or Xerox copies of each other as well as to the parent. And we use the term clones for such offsprings which are genetically and morphologically alike. So, in today's class, we shall proceed with asexual reproduction and we shall study the different types of asexual reproduction. The first one being binary fission. Binary fission is a type of asexual reproduction commonly found in prokaryotes. Once again, if the terms seem to be new to you, their meanings are written in the screen below. Okay. So this is a form of reproduction commonly found in prokaryotes, example bacteria and the archaebacteria, and some single-celled eukaryotes like amoeba and paramecia. Here, the parent organism divides into two to produce two genetically identical daughter organisms, two daughter organisms, and that is why this is known as binary fission. Binary fission takes place under favorable conditions. Favorable conditions, that means it would mean the suitable temperature, a sufficient nutrient availability or oxygen availability. Okay. Based on the plane of division, binary fission can be classified into the following types. Number one, simple binary fission, where the cell divides in any random plane. Example is amoeba. So if I have to draw the irregularly shaped thallus of amoeba and the darkly stained nucleus, the division of amoeba cell can happen in any plane, resulting in two daughter organisms. Secondly, is the longitudinal binary fission where the cell divides along its long axis. Example is euglena. So if I have to draw the euglena cell, okay, with its flagella, the division of the parent euglena occurs along the long axis, and this is known as the euglena binary fission, resulting in two daughter cells. Thirdly, we have transverse binary fission where the cell divides along the short axis. Example, paramecium. In case of paramecium, the cell division is transverse. And lastly is the oblique binary fission where the cell divides at an oblique plane. Example, serraceum. So if I have to draw the organism serraceum roughly with its flagella, the division is oblique. Here on your screen is shown to you the binary fission in amoeba. Here you can see the parent amoeba cell with an irregularly shaped thallus and a darkly stained nucleus. And the protrusion of the cell, the protrusion of the body is known as pseudopodia. Pseudopodia or false feet. These help in locomotion. Okay. Now, when the cell is ready for fission, is ready for reproduction, The first step is that it accumulates its cytoplasm towards the center. That means it pulls the cytoplasm, pulls the pseudopodia towards the center, withdraws, contraction of pseudopodia happens, and once the cytoplasm is densely accumulated within the cell, it is ready for karyokinesis. That means replication of the genetic material happens, and that genetic material is divided into two. The nucleus is divided, karyokinesis occurs. Eventually, cytokinesis, which means the division of the cytoplasm occurs, resulting in the formation of two amoeba cells. Point to be noted over here is that binary fission in amoeba or binary fission in bacteria, these are an exception to the concept of lifespan. In the previous class, we studied what lifespan is. Lifespan is defined as the time period from birth to the natural death of an organism. But in the case of amoeba, you can see over here that there is no concept of death of amoeba, because the parent amoeba is itself splitting into two, is splitting or getting divided into two cells to give rise to the daughter amoeba. The whole structure is getting divided. So the parent amoeba actually acts as a reproductive unit. It acts as a reproductive unit. And once binary fission has taken place, this parent amoeba, the cellular structure of the parent amoeba, is giving rise to two new cells, is giving rise to two new daughter amoeba cells. So we can say that binary fission in amoeba is an exception to the concept of lifespan, where the organism, in terms of biology, is actually immortal, because this parent amoeba is completely losing its identity to give rise to two new cells. It is losing its identity. There is a loss of identity. It's not that you are the person who is giving birth or dying. No, there is no concept of birth and death over here. Okay. Next, we study binary fission in euglena. On your screen, you can see the parent euglena. Okay. Similarly, in this case as well, there has to be the accumulation of cytoplasm with all of its nutrients, which is shown over here with the darkly colored green stain. And once the cytoplasm is densely accumulated within the cell prepares itself for the division of the nucleus. The genetic material is duplicated and the nucleus is getting split. That means karyokinesis occurs. The cell grows in size to give rise to two new daughter cells. Cytokinesis occurs along the longitudinal plane to give rise to two new daughter euglena cells. Thirdly, we study about the binary fission in bacteria. Now, reproduction in bacteria is a topic of great detail, and we shall go ahead with this topic later in the upcoming chapters. For now, you can understand that the dark brown color shows the cell wall of the bacteria, which is made up of peptidoglycan. The plasma membrane is in gray. You can see that the cytoplasm, and if in the cytoplasm, the genetic material, the highly coiled genetic materials is lying freely. There is no covering around the genetic material. There is no membrane around the genetic material, because bacteria are prokaryotes. That means they lack any membrane bound organelles. There is no organelle, membrane bound organelle in case of bacteria. So this naked form of DNA is known as nucleoid. Is known as nucleoid. Similarly, in this case as well, the DNA, the genetic material has to be replicated. Replication of DNA occurs. Okay. And you can see that with the replication of DNA, there is a furrow or a cleavage that is being formed. That means the division of the cytoplasm has taken ground. Okay. And at the cleavage, what happens is a contractile ring is being formed. This contractile ring is being formed at the plane of division of cytoplasm, and eventually the contractile ring contracts, squeezes out two new daughter bacteria cells. The next form of fission we shall study is multiple fission, commonly found in protozoans like amoeba and plasmodium. Now you might be thinking that why am I saying amoeba? We have studied binary fission in amoeba. See, when the organism is found to be in harsh climatic conditions or conditions unsuitable, unfavorable for its growth, maybe it could be due to a lack of nutrient availability. That time, these organisms they undergo multiple fission. Okay, and certain algae like Chlamydomonas and Chlorella, the nucleus of the parent cell divides several times by mitosis, producing several nuclei. Several. That is why multiple fission. The cytoplasm separates each of the nucleus, creating multiple daughter cells. So, two daughter cells nahi hai, there are multiple daughter cells. Like in the case of Plasmodium, this is the parent Plasmodium with its darkly stained nucleus. Immediately, the nucleus divides itself repeatedly into several multiple nuclei, all within the same parent cell. And eventually, each of this nucleus is surrounded by a covering, by a wall. Okay, and little cytoplasm forming daughter cells, multiple daughter cells, and that is why this is known as multiple fission. And when each of these daughter cells have accumulated some amount of cytoplasm and are enclosed by a covering, the parent cell breaks open to release the daughter cells. This plasmodium is the causative agent of malaria. Next, we study amoeba. Like I told you, amoeba undergoes multiple fission in the case of unfavorable conditions. Okay, so when conditions are unfavorable, this parent amoeba, you can see that the irregular shaped thallus is not there because it is preparing itself for fission. It has contracted its pseudopodia, pulled it within itself, and is ready to cover itself with a three-layered cyst wall, and this stage is known as incestation. Once it has covered itself, protected itself with these three layers of cystic walls, the nucleus of the amoeba cell, parent amoeba, repeatedly multiplies, repeatedly divides to form several nuclei. Again, each of this nucleus accumulates some amount of cytoplasm and a cell membrane to form pseudopodiospores. These pseudopodiospores are released by the parent amoeba into baby amoebae, which is known as amoebulae. Baby amoebae known as amoebulae, and these amoebulae grow and develop into mature amoebae cells. This is multiple fission and sporulation in amoeba. Okay. The next type of asexual reproduction is budding. It is a type of asexual reproduction commonly found in baker's yeast, scientifically known as Saccharomyces cerevisiae, corals, and some sponges. A new organism develops from an outgrowth or a bud on the parent organism. The buds eventually break away from the parent once they have fully grown into mature independent individuals. Let us have a look at budding in hydra. This is the parent hydra, and you can note over here a small outgrowth has been produced on the parent body of the hydra organism. Okay, now this bud accumulates nutrients, takes nutrients from the parent organism and grows in size. It grows and it grows further in size, it develops, and once it is fully grown and mature to survive as an independent individual, it detaches itself from the parent organism. It breaks away from the parent organism to give rise to a new offspring. Now, if you see over here, the identity of the parent hydra is intact. This parent hydra is intact when it is giving rise to the new offspring. So, the identity in this case is not lost. So, here the concept of immortality will not come. The identity is intact. We also study budding in yeast, and in yeast it is very important. I'll tell you why. You can see the parent yeast cell, nucleus, and a large vacuole. Once again, the nucleus, the genetic material, has to replicate, so we have two daughter nuclei. Okay, at the same time, a bud and outgrowth from the parent cell can be seen on the parent organism, and outgrowth is being formed. And once the nucleus has been duplicated, one of the nucleus is sent into this outgrowth, into this bud. The bud, along with its cytoplasm and nucleus, vacuoles, and all other cell organelles need to be duplicated and sent to the outgrowth. It grows and grows and develops to form a new individual, and it detaches itself from the parent yeast cell. A similar concept also takes place in certain fungal organisms. Okay, in certain fungi, the concept of budding takes place, where chains of buds are formed. Not one, but actually there are chains of these buds formed, like shown on your screen over here. These are many, many buds being formed. And these chains of buds are known as, it is known as the torula stage. Torula stage. Okay, and certain fungi like rhizopus and mucor produce asexual spores, produce these spores known as oidia, and these oidia function in the same way as the yeast buds, and that is why in them it is known as torula stage. These chains of buds that are being formed. The third type of asexual reproduction is fragmentation. Fragmentation is a type of asexual reproduction commonly found in Spirogyra, Rhizopus, planarians, annelids, etc. Annelids are segmented worms. Okay, they belong to phylum Annelida. Example is earthworms and leech, where a new organism grows from a fragment of the parent organism. The parent organism, it cuts into fragments. It is broken into fragments, and each of this fragment develops a new organism. Some plants have specialized structures for reproduction via fragmentation, such as gamete in mosses and liverworts. Example, Rixia and Markensia. Okay, we will be studying about Gemme and Gemma cups in detail in the next class when we study vegetative reproduction. Most lichens as well reproduce through fragmentation to ensure that each new individual contains both the symbionts, the alga and the fungal partner. In echinoderms like starfish, this method of reproduction is known as fissiparity. So if I draw a starfish and the starfish happens to cut one of its arm due to some injury, so this cut arm is able to produce a new organism. Okay, That means it is a form of regeneration. The cut arm, this is the, if this is the cut arm, the cut arm is able to regenerate a whole new organism. Okay. Here we study about fragmentation in Spirogyra. On your screen, you can see the parent Spirogyra. Okay. And it is known as Spirogyra because of its characteristic spiral chloroplast. There is the darkly stained nucleus, and each of the cell is divided by this septum. All right. Now, if I mark a point where necrosis, cell death, or injury is occurring, what happens is that the parent Spirogyra cell is getting fragmented at the point of necrosis. Okay, at the point of necrosis or injury, and each of this fragment will give rise to different three new individual organisms. So there occurs fragmentation in Spirogyra at the point of necrosis, and each of these fragments develop into new individuals. Then we come to spore formation. Plants and many algae undergo sporic meiosis, where meiosis leads to the formation of haploid spores rather than gametes. So typically, meiosis say we get gametes, right? Meiosis gives rise to haploid gametes, but in this case, we are getting haploid spores. And that is why this is not the typical form of gametogenesis. It is known as agamogenesis. The spores, when they disperse, they grow into multicellular individuals known as gametophytes without the occurrence of fertilization. So the meiotic spore, the spore is haploid, and without fertilization, there is no fertilization, it is giving rise to the haploid body, gametophytic body. These haploid individuals then produce gametes through mitosis. Okay, I will explain what this actually is happening over here. As you can see, the multicellular diploid organism which is bearing the sporophytic body. So if I have to show you over here, this is rhizopus. Okay, now the structure of rhizopus, this is the sporangiophore. That means it is the stalk bearing the sporangium. All right. So this is the sporophytic body of rhizopus, spore bearing body. Okay. Now this spore bearing body produces haploid spores by meiosis. Produces haploid spores by meiosis, like this one. You can see the spores are being produced, spores are being released. All right. Now these spores are being produced by meiosis, and the spores then fall on a suitable substratum and germinate to form the haploid gametophyte. They are forming the gametophytic haploid organism when they fall on a suitable ground. So the diploid body is undergoing meiosis to give rise to spores. These spores fall on a suitable substratum, okay, and give rise to the gametophytic haploid body. The gametophytic organism then produces haploid gametes by mitosis. These gametes undergo fertilization to form a zygote. Okay, the zygote develops into the diploid sporophyte once again. Now, as you can see, there is a alternation of the generation. Alternation. First, it starts as a diploid organism, does meiosis, produces spores. These spores germinate. Okay, does meiosis, produces spores. That means meiosis जैसे ही होता है, it enters the haploid phase of its life cycle. It germinates to give rise to a new gametophytic organism. This gametophytic organism by mitosis produces many gametes. These gametes, they fuse to form the diploid organism once again. This concept is known as the alternation of generations, and we will be studying this as well in detail. So, children, in today's class, we have studied about the different types of asexual reproduction. Previously, we went through what the meaning of asexual reproduction is, its advantages and disadvantages. In the next class, in the next video, we will be going through vegetative reproduction, which is a form of asexual reproduction in specifically occurring in plants. So if you have any doubts, any questions, any suggestions, please do leave it down in the comment section below. Thank you. Bye bye.